Welcome to this tutorial video. So as you can see in today's session, we'll be looking at bacteria. So before we even begin, I hope that you've subscribed to this YouTube channel so that you don't miss out on other upcoming videos. So to start with, we are looking at bacteria, and then I'd like to mention that maybe some of you may not be aware, if we say bacteria, that's a plural term. It simply means many. If we say bacterium, that is singular. So bacteria, that is many, a lot of bacteriums. So we'll be looking at bacteria, of course, in microbiology, and I just hope that you've subscribed so that as we keep on posting other subsequent videos, you are able to follow through. So why are we talking about bacteria, especially if we want to understand this kind of topic in medical terms? So basically, bacteria is one of the microorganisms that you're going to find in the universe. And basically, if you're talking about a bacteria, it's a microorganism. Simply means that you cannot see it with your naked eyes. Remember when I introduced microbiology in one of my video playlists, I talked about microorganisms being small in size and you cannot see them and you need a microscope for that. And a bacteria is one of the a bacterium is one of the microorganisms that you cannot see with your naked eyes. And then sadly, you find that these different bacteriums, they cause a lot of diseases and disorders in human beings. It is therefore important for you to acquire knowledge and understanding on how these bacteria, or how they grow, how they divide, how they can invade the human body, and what do they like and what don't they like. Because if you are talking about antibiotics in pharmacology, you are going to those pharmacists that make antibiotics, they first understand how these different bacteriums operate, how they function, and everything. So it's therefore important for you to acquire knowledge on this particular session. Okay, so as you can see here, I'm showing you this picture. This picture is trying to just remind you of something. That remember I said microorganisms are found almost everywhere on earth. Wherever you touch, wherever you sit, there are microorganisms everywhere. And then you can see that there's this particular picture which is showing this half to be the clean part, okay? Then this half to be a dirty part. So if the dirty part simply means that this part of the hand is not washed, okay? It's not washed or it's not clean, and then it has got microorganisms. Then this part of the hand which is washed, it doesn't have microorganisms, specifically bacteria in this particular case. So it's important, it's reminding you that washing your hands is one way that can protect you from having this particular bacteria. Okay, so let us make progress. Now, in the introduction, we're saying that bacteria are single-celled microorganisms that are incredibly diverse and found everywhere on Earth. So, if you're talking about bacteria, remember, if you're talking about major classification of living organisms, we use what we call prokaryotic cells and eukaryotic cells. Now, bacteria falls under what we call prokaryotic cells. They contain only one single cell. So, no one was saying that bacteria are single-celled microorganisms that are incredibly diverse. It simply means that they are in different shapes, they are in different forms, they exist in different ways. So, these kind of bacteria that we're talking about, they have got different forms, and there are a lot of types that we're going to talk about. And then they're found everywhere on the Earth. So today, bacteria are considered one of the oldest life forms on Earth. These different bacteria we're going to talk about, they have been in existence for a long time ago. Okay? When I was saying that they are considered to be one of the most oldest form of life on Earth. Now, most bacteria make us ill or cause diseases. Most of these bacteriums that we're going to talk about, they make us ill. You find that maybe you're coughing, maybe you've got flu and everything. Some of these bacteria are responsible for making those kind of or bringing about those kind of diseases or illnesses. But it's important to know that they have a very long mutual relationship with humans. So in as much as this particular bacteria causes disease or illness, they still have a relationship with the human beings. Why? Because not all the types of bacterium cause disease. We're going to talk about that. So in this particular session, we're going to explore important aspects that you need to know about the bacteria. The general objective is that at the end of this tutorial video, you should be able to acquire knowledge on the bacteria. Then the specific objectives are that one, you must define the key terminologies that are used in the description of bacteria. And then you should also be able to describe the general shape of a bacterium, because you understanding the general shape of a bacterium is also one way that medical personnel that we can use to be able to classify bacteria, going about the approach of treatment, what kind of drugs are we going to give this patient and everything. Sometimes we can use the shape of the bacteria to determine those kind of things. Apart from that, you should be able to outline the main classifications of bacteria. Remember, in the introduction I said that bacteria are diverse. They are in different forms, they are in different shapes. So it's important for you to find a way of how to classify these different bacteria. Otherwise, if a patient is sick, you wouldn't know what to do. You don't know what kind of medication I'm supposed to give. But based on the classifications, you're going to find it easy, because when you're classifying something, you're just grouping them in forms or ways which are similar. So we're going to look at all these different aspects. So let's start with the first objective. Talking about the definition of terms. Maybe you would like to know what is a bacteria. Actually, it's supposed to read what is a bacterium, because bacteria is plural, bacterium is singular. So a bacterium or bacteria are a diverse group of microorganisms. They are microorganisms. They are a diverse group of microorganisms that are prokaryotic in nature, meaning they lack a true nucleus and other membrane-bound organelles. So basically, if you're talking about bacteria, these are diverse group of microorganisms that are prokaryotic in nature, meaning they don't have a nucleus. Additionally, they also contain only one single cell, okay, and they lack other membrane-bound organelles. For example, if you're talking about a nucleus, bacteria do not have a nucleus. A nucleus is a membrane-bound organelle. So there are some organelles that have got a membrane, like the mitochondria, like the nucleus, have got their own membrane. But if you're talking about a bacteria, it's only a single cell that is lacking a nucleus, that is lacking the mitochondria and other organelles that you're supposed to find in a normal cell. So a bacteria, these are microorganisms that are prokaryotic in nature, or that lack a nucleus. And then when we say prokaryotes, prokaryotes simply means that cells lacking a true nucleus, we call them prokaryotes. And all bacteria, all of them, they are prokaryotes. They lack a true nucleus. Apart from that, there are these simple terms that you must be familiarize yourself with as you talk about bacteria. One, DNA. So maybe you just know that DNA is only found in human beings and everything. Even bacteria has got DNA. Bacteriums do have DNA, because they can also multiply, they can also divide, they can also increase in size and number. All right. So they contain also what we call DNA. So DNA simply means deoxyribonucleic acid, and then they also have RNA. Why? We are telling you about these things because bacteria also produce what we call proteins. The same way human beings perform protein synthesis, even bacteria are also able to do that. So it's important that we remind each other about what DNA means, what RNA means. So RNA simply means ribonucleic acid. Remember, these are different forms of molecules that contain genetic information, either in form of DNA or RNA. Let's make progress. Then the second objective was that we're supposed to look at the structure of a bacterium. The structure of a bacterium. So you can see here, this is the general structure of a bacterium. Pay attention, because sometimes in exam questions or papers, they will require you to draw the structure of this particular bacterium, and maybe they also ask you to talk about each and every function of each part of this particular bacterium. So to start with, this is the general structure of a bacterium. So this is how a bacterium looks like. And like I mentioned earlier, that these different bacteria, they exist in different forms or type. So you may find that others, they may not have this kind of flagellum, for example. We're going to look at those things. But generally, this is the general structure of a bacterium. So let's start looking at it. So you can see that this bacterium, like we said, it is a single cell. It only has one cell. Just imagine a cell having a tail-like, which we call a flagellum. So it's just one cell that you can see here. So a bacterium has got what we call a flagellum. This, okay, this sweep-like structure here. This is a flagellum. So a flagellum, basically, it is used for locomotion, for a bacteria to move from one specific part of the body to the other and everything, it uses a flagellum. It's the one that propels it, or it's the one that helps this particular bacterium to what? To move. And then, apart from that, because we said a bacteria is just like a cell, but a single cell. So let's just zoom in here and look at something. So you can see that this particular bacterium has got what we call a capsule. Okay, a capsule. So maybe some of you have come across a tablet pill, which we call as a capsule. Then inside that particular pill, it contains medicine inside. So the reason why they put that particular medicine content inside the capsule, they just want to protect the medicine inside. Even a bacteria has got what we call a capsule. So this capsule is just a structure that protects a bacteria from hazards or environmental hazards outside the environment. So you find that the actual bacteria is inside, then it's surrounded by what we call a capsule. So this is the structure that is protecting a bacteria. Then after you move from the capsule, start going inside, there's what we call the cell wall. So the cell wall is also found in bacteria. I know that maybe you all know that the cell wall is found in plants. Even in bacteria, you find the same cell wall. Okay? The same cell wall. Apart from that, if you now go inside, you find that there's what we call the plasma membrane. So we said we've got the capsule, then we've got this cell wall, then this yellowish like line here is what we call the plasma membrane or the cell membrane. So even the bacteria has also got the cell membrane because it also wants to regulate what is entering inside it and what is leaving. Apart from that, just like you know, it also has the cytoplasm, the jelly-like substance that is surrounding different parts or components of this particular cell, the cytoplasm. So it also has the cytoplasm there. Apart from that, it also has the ribosomes. Okay, so maybe I didn't say anything about the cytoplasm. If you would like to know, the cytoplasm of this particular bacteria, remember the bacteria is only having one cell. So this cytoplasm is the one that contains a lot of enzymes that are used in metabolic processes for this bacteria to digest things or use them for energy, or for it to be able to have things that it can use for energy. It uses enzymes that are contained in the cytoplasm. Okay, then talking about the ribosomes specifically, they help a bacteria to form proteins in protein synthesis. Just like I said that in human beings you've got RNA, you've got DNA. Even a bacterium has got DNA and RNA, which helps it to synthesize protein. So bacteria also uses ribosomes in protein synthesis. Apart from that, there is this particular structure that we call a plasmid. This is very, very important. If you are a student that is having this course of microbiology and then you're talking about bacteria, there is what we call a plasmid. Now, before I even talk about the plasmid, just hold on a bit. Let's talk about this. So I hope you've been able to see this coiled-like structure here, which we call a nucleoid. A nucleoid and a nucleus, they are not the same. A nucleus is a structure, it's a membrane-bound organelle that protects DNA inside a human being, for example. But we said bacteriums do not have the nucleus. So they only contain this particular DNA molecule, which is just coiling outside. It's not protected by anything. So collectively, we call it as a nucleoid, not a nucleus. It's a nucleoid. So a nucleoid is basically a DNA containing material. Of course, this is the material that contains genetic information. Apart from that, then I talked about you knowing what we call the plasmid. So the plasmid, as you can see, it's similar to the DNA or the nucleus, but it's separate from it. It's just alone. It's independent. So a plasmid, this is the poisonous part of a bacterium. It's the one that makes a bacterium to be resistant to what we call antibiotics. For example, if you are coughing, you are sneezing, they give you medicine, and then the medicine is not responding. They give you tetracycline, they give you amoxicillin, nothing is happening. It simply means that most likely the plasmid part of the bacteria is the one that is resisting that particular medication. So it's important for a healthcare provider to understand that a plasmid is a structure that is resistant to antibiotics, especially if you are treating a patient who has got bacteria. Okay? So you find that some bacteria are resistant to antibiotics, like, for example, tuberculosis, Mycobacterium bacillus. It's resistant to antibiotics. So plasmids are one of the structures that contribute to a bacteria being resistant to antibiotic drugs. Now, let's also not forget that we've got these projection like Okay, we've got these hair-like projection that you can see here. We call them the pillars. We call them the pillars. So they're in different forms. I'm going to show you another picture very soon. They're in different forms. So there's what we call the pillars. So if you come across a term that is being referred to as the pillars on a bacteria, so the pillars, they are not used for locomotion at all. These are specifically used for sexual reproduction. Okay, for sexual reproduction, maybe you're wondering to say, oh, bacteria only uses it as sexual reproduction. Yes, I understand, but there is a concept which we refer to as conjugation, in which a bacteria shares its genetic information to other bacterium. So the fact that there's contact between the pili and another bacterium, we call it sexual, kind of reproduction. I'm going to explain. So we call them the pillars. They give, they're the ones at which we say they give male-like structure of the bacteria. So if a bacteria has got pillars, we're going to say it's a male-like structure. It's a male-like type of a bacterium. Now, there's also something that I want to show you. So there's this another picture here which has got everything that I've talked about, but the difference is that you can see that there are these pillars. Okay, so pillars is singular, pillar is plural. So you can see that we've got this particular pillar structure. Now these pillars, this pillars here is the one that is used for what? For sexual reproduction. And then there are what we call the family. So this is the family. The long ones that you can see here, these long ones projection, are what we call the pillars. I said these are the ones that you use, that the bacteria uses in sexual like production. Then these small short like hair like projections, we call them the family. These are the ones that allow a bacteria to adhere to body surfaces, to internal organs inside the body, or to stick to body surfaces or internal organs inside the body. We call them the family. But the pillar is it's a bit longer than the family, and this one I've said it's basically used for what? Sexual reproduction. Of course, this is the flagellum, this is the plasmid that we talked about, the nucleoid, the ribosomes, and the capsule, as well as the cell and the cell membrane, as you can see. Just the same diagram. It's just that this diagram didn't, the previous diagram didn't have what we call the family. So I wanted to show you the difference between the family and the pillars that you are seeing there. So this is the structure of the bacterium that you may be required to draw or maybe even label in case you are being asked a question to be able to do so. Now, talking about the specific functions. So in case maybe we didn't get the function that I said, these are the functions of the different parts of the bacterium that we talked about. Remember, it's important for healthcare personnel to understand each and every structure of a bacterium, because when drugs are being made or when drugs are being administered, they target specific structures of this particular bacteria. So other drugs, maybe they're going to target the capsule, other drugs, maybe they're going to target the cell wall, others are going to start to target the nucleus and everything. So it's important for you to at least understand if this capsule is there in a bacteria, then what does it do? And if I give this particular medication, is it going to kill this bacteria or not? So for example, on the capsule, a capsule is made up of what we call polysaccharides. You know polysaccharides, right? Combination of different glucose molecules, putting them together, 3 or more, then you'll be able to form what we call polysaccharides. It helps the bacterium from drying out and protecting it from phagocytosis. So these polysaccharides, they keep a bacteria to not be able to dry out, okay? And then they also protect it from phagocytosis. Remember, phagocytosis is a process in which white blood cells kill and engulf microorganisms, especially pathogens. So you find that this capsule is protecting the bacteria from these white blood cells. And then this bacteria is going to have a long living life inside your body in case it has infected you, because it has got this particular capsule that is protecting it. Apart from that, I said the bacteria has got what we call the cell wall, okay? This particular structure. So we've talked about the capsule, then this is the cell wall. This is the cell wall here. From the cell wall, we'll talk about the cell membrane. Then the cytoplasm and each and every structure that we're going to, refer to here. So the cell wall, it encloses the bacterium with what we call peptidoglycan. Now, you can see here that I've even labeled this or indicated it in bold. So a peptidoglycan is a very, very important structure in a bacterium, not just in a bacterium, but also those that create these pharmacists that create, medicine and everything, because we can classify a bacterium based on its peptidoglycan layer. I'm going to explain very soon. So a peptidoglycan, what does it mean? Peptido meaning they are proteins, glycan, glucose. So the cell of a bacteria has got what we call peptidoglycan, which is just basically a combination of a protein and the sugar-like molecule. The cell wall gives the bacteria its shape and surrounds the cytoplasmic membrane and protecting it from the environment. So the cell wall of this particular bacteria, it's the one that gives the bacteria its shape, also protecting what? The cytoplasmic membrane, the plasma membrane, as well as the entire content inside the bacterium. Apart from that, it also has what we call the cell membrane or plasma membrane. This is a double layer structure comprising of lipids and proteins. So if you're talking about the cell membrane, that cell membrane is made up of lipids and the proteins, okay, which act as semi-permeable membrane for uptake of nutrients through passive or active diffusion. Remember I said, just like a human cell, or an animal cell, even a bacteria has got the plasma membrane, which regulates the entry and exit of materials. You don't want anything to enter inside or anything else to leave. So this plasma membrane is the structure in a bacteria which contains the lipids, the fats, right? And the proteins, and it's also regulating what must enter and leave the bacteria. Apart from that, we also talked about it having what we call the nucleoid. Remember the nucleoid, not the nucleus. So the nucleoid, this is a region of the cytoplasm. This one that we talked about here. Let me show you. So this is a nucleoid. It's a region within the cytoplasm that contains, of course, DNA. So this is a region in the cytoplasm that contains chromosomal DNA, and it is not a membrane-bound nucleus, but simply an area of the cytoplasm. Remember, the difference is that in an animal cell, you find the DNA being enclosed or being protected by the nucleus. However, in a bacterium, the DNA is just outside, everywhere, inside the cytoplasm. That's the difference, and we call it a nucleoid. So, like I said, the nucleoid contains this DNA, and this DNA is the one that contains genetic information of the bacteria. So some drugs, what we're going to do is that when you give a drug to a patient, that drug is going to target the nucleoid, which contains DNA. If you kill the DNA of a bacteria, most likely you can kill the entire bacterium. Apart from that, we said it also contains the cytoplasm. So the cytoplasm of a bacterial cell is where functions of the cell, such as cell growth, metabolism, and replication are carried out. Remember I told you, the cytoplasm, this is a jelly-like liquid, environment which is responsible for different things. So for a bacteria to grow, because for a bacteria to grow or increase in number, there should be what we call DNA replication. Then there are enzymes inside this cytoplasm that must do those particular pathways or processes. So this cytoplasm facilitates or makes sure that there's cell growth, there's metabolism, there's also what replication inside the bacteria. Apart from that, I talked about the ribosomes. So these are, the bacterial ribosome, this is basically the cytoplasmic nucleoprotein particle whose main function is to serve as the site of mRNA translation and the protein synthesis. So mRNA simply means messenger RNA. Translation is a process in which now you are about to start forming proteins. Okay? You are converting mRNA to specific amino acids, of which those amino acids now, if you group them together, they will form what we call proteins. So now that I said ribosomes, these are structures in a bacteria, just like they perform the same function in animal cells. They also help a bacteria in what we call protein synthesis. Even a bacteria needs proteins. Okay? Apart from that, I talked about the plasmid. Remember I said the poisonous part of the bacteria, or the most toxic part of the bacteria. So a plasmid is a small circular double-stranded DNA molecule that is distinct from the cell's chromosomal DNA, because we said there's a central DNA molecule which is in the nucleoid, then there's another extra DNA. So it's like a bacteria's got two DNA. The main one, which we call chromosomal DNA, and that extra one, which we call the plasmid. Often, these genes carried in the plasmid provide bacteria with genetic advantages such as antibiotic resistance. I said that this plasmid helps a bacteria to resist antibiotics. So you keep on pumping tetracycline, amoxicillin, penicillin G. different antibiotics, but you find that the patient is not getting better. Why? Chances are they have got a bacterium which has got a plasmid. Then this plasmid is resisting the effects of that particular drug. So it's very, very important that you know what this plasmid is doing in a bacteria. Apart from that, I talked about what we call a pili. A pili is plural, pilus singular. So many species of bacteria have what we call pili. Okay, so singular is pilus. Small like hair projections emerging from the outside of the cell surfaces. These they assist the bacteria in attaching to other cells and surfaces such as teeth, intestines, and the rocks. But to be more specifically, if you are talking about pili, because other books they will just say pili, they will not tell you to say this pili exists in different forms. So there's what we call ordinary pili, or what we call the family, the one that I've shown you. So the family plays a role in adherence of the bacteria to host cells. It helps the bacteria to attach or to stick to host cells. We call it the family. Then there's what we call the sex pili, those long ones that I showed you. Okay, so the sex pili, it gives the maleness of a bacteria. So during bacterial conjugation, the male donor or the male donor cell adhere with its sex pili and transfer DNA to a non-male cell, which we call a recipient. I'm going to show you. So this bacteria, if it wants to make another bacteria to be also poisonous like itself, it's going to share genetic information through what we call conjugation. I'm going to show you. So that kind of bacteria with a sex pili, the one which is sharing the toxin part, is the one which we refer to as the male one. So this is what I was talking about. So for example, there's what we call bacterial conjugation. This is where now a bacteria wants to share genetic information to another bacteria which is not toxic. Let me give you an example. Let's assume that you are infected with this bacteria, and then it wants to make other bacteria inside your body to be toxic like itself. Let's say these ones on the left, they are not toxic. Then these ones these ones on the I think this is your right if I'm pointing like this. This is your left if I'm pointing like this, yes. So these ones on the left, they're the ones, this bacteria on the left is the one which is toxic. These ones on the right, they are non-toxic. So if a bacteria wants to make other bacteria to be toxic as well, it performs what we call bacterial conjugation. So what it's going to do is this. Remember, this is the bacterial cell, right? It has got chromosomal DNA or the nucleoid here, then it has got a plasmid. If you look at this other one, it's innocent. It doesn't have a plasmid, meaning this one can't resist antibiotics. But what a bacteria is going to do is that it's going to share this information from the plasmid, giving this one so that it also becomes resistant to antibiotics. We call that as conjugation. And then for it to do that, it uses what we call a sex pili. So a sex pili from the donor, which is labeled as F+, from a donor, is going to attach to this one, a recipient, which is labeled as F-, and then they're going to attach here. Okay, they're going to attach there. If they attach, there will be transfer of genetic information from this donor cell to this recipient cell. As you can see, this plasmid is also being transferred from the donor cell to the recipient cell. As a result, this one also has the new plasmid. It has also become resistant to antibiotics. So that is what I meant by bacterial conjugation. Okay, apart from that, we talked about the flagellum. So the flagella is plural. Singular, we call it flagellum. These are hair-like structures that provide a means of locomotion for the bacteria that have them. So they can be found on either, or they can be found on one side or both sides of a bacteria. We're going to talk about that. So these are the structures that you need to know on the bacteria and what they do. Okay? So in the next particular video, we'll start talking about how we can classify bacteria based, for example, on the number of flagella, based on the shape, okay, based on what you call gram staining. So make sure that you subscribe to this YouTube channel so that you don't miss out on that particular video. I hope this has been able to help you in some way, and ensure that you hit that subscription button so that you don't miss out.